Good, thank you very much. So, yeah, well, thanks for inviting me to this excellent webinar today afternoon. So, this is going to dovetail from some of the talks that we've heard in the last couple of sessions, but also give a slightly unique sort of look into what we're trying to do in the non-monogenetic rare disease space, particularly with reference to rare liver disease. Now, I'm a clinician. My clinical job is very much looking after people with rare liver diseases. But as a clinician scientist, I have a big interest in trying to leverage large-scale administrative healthcare data to better understand the outcomes and clinical journeys that patients experience, the diversity in care, and to find out where some parts of care in the UK are not necessarily reaching set standards that we would like according to either guidelines, consensus statements, or indeed a sort of evidence-based practice where we have that, and also how we can modify those. Now, today I'm actually going to show a couple of examples of how we've used administrative healthcare data to better understand the natural history of rare liver diseases, but also how to improve clinical trial recruitment and also clinical trial design and facilitate early market adoption of orphan drugs. So just a little bit about liver disease. Now, some of you may have seen a variation of this before. It's a little bit old now, but it's from the Lancet Liver Commission. What it shows is that liver diseases as a whole are the glaring exception when it comes to sort of mortality compared to other chronic diseases that we see in adults. When you look at mortality from things such as diabetes, from heart disease, from strokes, etc., the rates of those things are going down, but for liver diseases, they've been rising and rising and rising. Now, there is a slight dip with the advent of chronic viral hepatitis therapies, sort of about 10 years ago, but that rise is continuing higher and higher. Now, mostly, this is not due to rare disease. Mostly, the deaths that we see in people from liver disease are amongst those people who drink too much or have metabolic risk factors such as obesity, diabetes, hypertension, hyperlipidemia. That's the lion's share of liver disease that we'll see in secondary care. However, when we talk about sort of the burden of liver disease in the UK, it is not just about mortality. I work in a large liver transplant program here in the West Midlands, and what we see is that the most liver transplants are actually performed for people with autoimmune liver diseases, which are rare conditions. Now, it is a little bit of a cheat code, so the arrows move slightly, but the three autoimmune liver diseases that lead to transplantation are PSC, which is called primary sclerosing cholangitis, PBC, primary biliary cholangitis, and AIH, which is autoimmune hepatitis. Now, these are conditions where, sort of much like other rare conditions, we don't have a cure for them. For PBC and for AIH, we have good medicines that can maintain disease, disease state, or allow people to, on medicine, lifelong medicine, to enter a state of remission, but we can certainly not get rid of it. And PSC, which I'll talk about first, is a real black box of hepatology, because we don't know why people get it. We have no real robust way of predicting who's going to do well and who's going to do badly. And more than 50% of people will end up needing a liver transplant or sadly dying from their liver disease on average, between 14 to 20 years from first presentation. Now, how have we used healthcare data to better understand the clinical course that patients experience? Oh, before I continue, it's just to highlight that because these are autoimmune diseases, about 1 in 3 patients do develop recurrence even after liver transplantation. And this is one of the biggest drivers for why people lose their liver grafts and require retransplantation. So the journey of these patients doesn't end just because we put a liver transplant into them. Now, when it comes to PSC, to try and understand some of the comorbidities associated with it, because we tapped into HES data, right? Now, HES data has its own problems, but it has many strengths. And using validation methods from our own center to try and develop an algorithm where we can accurately identify patients, we were able to show that 70 to 80% of patients with PSC also have a form of inflammatory bowel disease. Most often, this is picked up before the PSC is diagnosed, but quite a large number of individuals are diagnosed around the same sort of time that the PSC is picked up. When we look at people with inflammatory bowel disease, whilst we do see people with primary sclerosing cholangitis, in about 5%, a much larger proportion of these individuals were actually found to have subtle radiological changes. This is a disease that affects the bile ducts of the liver. These people were found, even with normal liver tests, to have very subtle bile duct changes, indicating that the disease may be more common in people with inflammatory bowel disease than we originally thought. Now, to try and find out where these people are, because if we want to access people for clinical trials, if we want to use sort of healthcare data to enhance trial recruitment and design, we need to understand epidemiology. Now, using whole of England data, sort of the University Hospital's Birmingham informatics team did this very nice piece of work with us. And what they found was that most of the PSC and inflammatory bowel disease cases tend to cluster towards the Midlands and the North, whereas most new onset inflammatory bowel disease cases actually sort of were more common towards the more southern parts of England. The second thing that we were able to do was capture a wealth of clinical outcomes to identify phenotypic factors that were able to heighten risk of disease progression, or but also reassure some people that they may have a more sedentary clinical course. Now, how is that? The point number one, very simply, this has allowed some degree of enrichment when we recruit people to clinical trials, because this disease has a very slowly progressive clinical course. As I mentioned, it progresses over years and years, not weeks or months. So when we're designing a clinical trial, it is not economically viable to recruit people into an interventional study of that longer period. So you want to enrich for your population who are more likely to experience clinical events over a shorter period of time. So we recognize that this particular condition predominates in people of young presenting age, and it's the younger individuals, and when I say younger, I'm referring to people who are 20s, 30s, 40s, people sort of either in higher education, starting their families, buying their first homes. It's those people where it really sort of affects, and that's why we try to enrich our clinical trials for that participant population. We recognize that there are different types of inflammatory bowel disease, which is indicated at the top of this slide. And whilst the incidence of each type of inflammatory bowel disease alone is relatively stable in the sort of across England, that of primary sclerosing cholangitis is actually rising year on year. So whether that may be due to better diagnostics, increased awareness of the condition, or a true etiological risk factor is what we're trying to unpick. Now, I mentioned that when it talked about the natural history of PSC, that it's a very slowly progressive clinical course. So the median transplant-free survival, as I sort of indicated, is around about that 14-year mark. So when we see people presenting it sort of with a milder clinical phenotype, clearly they have sort of longer before they run into clinical problems. But to try and unpick at least a suitable biomarker that we could use, a surrogate that we can use to show that drugs are working, and also to try and identify genetic factors associated with disease development and disease progression, the UK PSC registry was founded. Now, and this is, this is set up as an observational clinical trial. This is prospective, consent-driven across pediatric age groups and adult age groups, and it involves all the 4 nations sort of the UK. And just sort of the turn of the year, when we looked at how many people, we've sort of about 3,500, just over that number, have been invited to participate in the study since it was originally set up in 2008. And we've got just over 2,400 registrants, predominantly adults, where we capture clinical data and annual event updates. Now, we've also created a biobank for these sort of patients for serum and DNA, because I mentioned sort of we are trying to identify biomarkers associated with disease progression. This was very much a later thing. Initially, it was very much to capture the clinical course of patients. Now, we do consent for serial sample measurements, but because of funding constraints from following the initial grant, we haven't yet managed to get serial samples to look at delta changes and stability, but at least it's a starting point. And we ask every centre in the UK, and all hospitals are eligible to take part. We ask for an annual update of all cancers, all forms of bowel surgeries, liver transplantations, and if patients have sadly passed away, what they have passed away from. Now, this is all well and good at getting nice papers to show that we understand the natural history of what UKPSC, of this, patient population, and it's good at trying to identify certain biomarkers that we can use as surrogates. But when it comes to designing clinical trials with rare disease that have got this long variable clinical course, and how can we leverage this data in a slightly better way? In interventional studies in PSC, and indeed some of the other rare liver diseases that I spoke about, generally speaking, even big commercial studies such as those from Gilead or GSK and certainly some of the big pharma, they're not likely to run more than 2 to 4 years at a time. I think the largest trial that I've sort of taken part in this particular patient group, or indeed any of the other autoimmune diseases, has lasted for about 4 years when it comes to the double-blind treatment phase. So you're locking in people to potentially a placebo group for that long period of time, which certainly according to our patient and public involvement groups is unethical. And we say to them, and quite rightly, they understand that every new medication has risk. But if we're trying to explore new or repurposed medicines where we know the safety and toxicity profile, we are still asking them to enroll into a clinical trial for that long period of time. And when we go to regulators, they say, well, you can't use a natural history data unless it's systematically collected as if you were doing it into a clinical trial. We need to know every single thing that happens to them, all their con meds, all their comorbidities, any adverse events that, for example, if they present to their local hospital in Slough or Bristol, and what the reasons are that they're presenting, what about other disease flares that are actually taking part. So to do that, we've started to now link our UKPSC registry to various NHS digital platforms. So that will enable us to capture data around various different medications that they may start that may or may not be related to their liver disease or their bowel disease, and also trying to develop healthcare cost of illness models, which are important when we're trying to justify to NICE and other bodies for health technology assessments of newly implemented therapies. And that's something that we haven't been able to do before. Now, it may sound like very pie in the sky thinking, saying it's all well and good. You want to link your registry to NHS Digital to capture every single part of the patient journey. And you've consented your patients to do that. But will it work? And if so, what can you do with it? Well, I'm just going to talk about, in the second part of this talk, about PBC, which is a disease that also affects the bile ducts of the liver, but more so affects people, women above the age of 50 years old. Now, with PBC, we do have medicines, and very recently we've had big randomized control trials that have been published, showing that they have biochemical efficacy, they can improve liver biochemistry, but these have been granted only conditional market approval. And a bit like PSC, this disease progresses over many years and decades, and it's not viable to conduct clinical trials of these drugs for that longer period of time. But the FDA and EMA, from when we were in the EU, said doesn't matter. We need to have some evidence that it's not just improving your liver blood tests. We need to have some evidence in PBC that these particular medicines are actually improving long-term transplant-free survival. But the problem is, when you've got a drug that's been granted conditional approval, and then you're then asking the same patient group to take part in a placebo-controlled trial, then that's not going to be deemed ethical. And as you can see here, this is a slightly busy graph, and I trust me, it took a long while for me to understand it when our statistical colleagues gave it to me. But if you focus on the pink panel here, what it shows you in this curve that looks like a Kaplan-Meier plot is the incidence of people dropping out from the placebo group and going on to commercially available obeticholic acid. And you can see that the time that they took to do that is between 1 to 2 years. Now, we did see people who were in the active treatment group also drop out, but you can see that the 25% mark was actually much, much later on. So this is challenge number one. It was not possible to recruit to this multi-million-pound trial because of the dropout rate of patients rolling over into commercial therapy. So then we went back to the FDA and we said, well, look, you preach about natural history studies for drug development. You tell us that we can use our databases, and then as well as having a UK PSE and a UK PBC registry, we've got a global registry where other centres have tried to follow what we've done here, and how can we leverage that piece of work to try to accelerate sort of drug approval for this, or full approval, for this disease. Well, they said you need to tell us what's happening to your patients. You need to sort of capture as many clinical events as you can, not just related to the disease, but also when they start other medications, such as off-label therapies that some people are doing. So to do that, sort of, we put our UK resources together with the global PBC sort of group resource, and we said we've got people who were in the original registrational trials where biochemistry was the endpoint, and they are now rolling into a long-term open label extension phase. And we've got our UK and we've got our global PBC study group sort of data where people who may have been eligible but not received therapy, we can use those people as external controls. And they said, well, why have they not received therapy? Are you going to have anybody who is eligible for therapy but not received it? So then we had to go back to the UK and we said, well, look, let's do a national study. And sort of long story short, what we found here, sort of in this nationwide audit of care delivery, is that less than 50% of people who were eligible for receiving this medication actually got it. So we had a good source control, sort of source data for using as a control. So what we then did was use external control data, and we then sort of propensity score matched them to the people who took part in that clinical trial, who were then taking part in an open label long-term extension, and balanced the cohorts as much as we could statistically. And it's not, and again, matching is never as good as a randomized control trial, but this is the best that we could do. And what we were then able to show was that certainly in the global and in the UK groups alone, that people who took part in the POISE trial, which was the obeticholic acid trial, had very few clinical events over several years compared to external control patient populations who were of a very similar ilk. Sort of justifying that we can actually use this therapy. Now, the good thing about sort of using these sort of external control group is that it does, in some way, give people an out when it's difficult to recruit to clinical trials of rare diseases with a long clinical course. But the other thing, just to finish off, is that now by linking UK PBC data to NHS Digital, what we've been able to do is identify people around the country who registered to receive information from when there's a new trial that's eligible for them. And so whenever there has been a new trial that's eligible for them, we can write to their local PI and we can say, dear Dr. Smith, you have these 10 people who you very kindly recruited to UK PBC. Through linkage, we've identified that they are eligible and we pre-screen them across the criteria for this trial. Could you please make contact with them? And if they're interested, please allow us to sort of, please approach them and send them to their local trial centre. And as a result, the UK has managed to recruit to PBC clinical trials more so than all our colleagues in Europe combined, and indeed in centres in North America combined, as represented by these, these on screen. So we're hoping to, PBC was very much a beta test, PSC is what we're trying to do now, and we're hoping to roll this out through a broader array of rare diseases where we've already got existing registries that we can apply linkage to, particularly those with active clinical trials. So hopefully I haven't gone through that too quickly and too much over time.